multiplexing. So like I said, there's an adapter on either end of the fragment. And what you can do is you can basically say, I'm going to use a different one of these 224 things. So one of them is Illumina TrueSeq adapter number 1, and on the other end is Illumina TrueSeq adapter number 2. And so that basically increases the resolution of your barcode, so you could have, in theory, 8 times 8, or 24 times 24 different possible things. At most, we've generally only, I think Illumina really only recommends going up to 96 samples. So like I said, that's basically come out to 1% of your reads per sample, and there's going to be some variability. We actually recently had a very specialized run where this one group that was collaborating with UNC wanted 900 samples per lane. It was kind of a crazy request, but it sort of worked in their experimental. It was enough for what they wanted to do in terms of experiments. And after a little bit of work, we managed to figure it out. And yeah, I mean, each of them was like 0.1%, but that's what they wanted. So we managed to get that to work. So that is most of what I have to say about Illumina sequencing. Are there any questions or okay? So it's sort of, there are a number of things that are sort of somewhat understood or sort of have been noticed about the Illumina sequencing. One is that, especially if you're doing like whole genome sequencing, there are parts of the genome that sometimes Illumina just kind of skips. Sometimes it's sort of repeat regions, sometimes it's other things, but when you're doing sort of genome assemblies, they're definitely, when you look at the coverage, like if you get something that should be 50x coverage of the genome, you notice some places get better coverage with Illumina, and other places get a lot less coverage. It's not entirely understood why that happens. There have been, and these have shifted a little bit over time as Illumina has changed their chemistry in terms of how things flow through, and like people will sort of do some statistical analysis and figure out some bias, and then Illumina will go through and change it, and then they'll basically introduce some new bias. There used to be, it's a lot less of a problem now. It used to be that some of the lanes were not considered as good as other. The 2 outside lanes used to not be considered very good. I think it's because at the time the optics weren't as good, and so the resolution of the laser and the camera that was looking at it could not see the edge lanes as good. But that has pretty much sort of disappeared as far as I know. It's still sort of generally, I think, considered the case that you generally want to do 2 biological replicates of anything you sequence. That was especially true of a lot of stuff when people were doing ChIP-seq experiments. You make 2 preps, you run each prep twice through the machine. That, is sort of a way to make sure that a particular run was not good, both on the sense of your experimental preparation and in terms of running it on the machine. Okay. Yeah, so let me repeat that question for the people online. So the question is, do you flow all 4 nucleotides at a time and then image? So yes, all the nucleotides are flowed at the same time. Like I said, before you start the point where you're actually taking the camera images, you first go through the state where you simply build up the colony. So yes, you're always sort of doing a flow through the thing. And they have this thing where there's a terminator. So the way you can actually do it in distinct cycles is there's a terminator cap on these nucleotides so that only one will be incorporated during a flow. Then they have a different reagent flow that allows it to then incorporate the next. Right? So it basically primes it for another nucleotide, and then the second flow comes through that has all the possible nucleotides. So each one of these colonies is built up 1 base pair at a time during a cycle. So you flow through all the nucleotides one of them gets incorporated, it's sort of capped off. Then another flow comes through to remove the cap, and then you can flow another thing to build them up one at a time. And like I said, once they've gotten to a certain size, then you actually go through and image it. Right? Good. Any other questions? Yes? Okay, so the question is, how do you know how much to sequence, basically, how many lanes and stuff? You sort of almost answered your own question. It basically depends on what study you're doing. Like, especially for sort of like metagenomic stuff or something. I don't know the specific numbers for things, but basically you have to check the literature, right? Has anyone done something like this before? How much sequencing have they done? I mean, you make certain estimates basically based on your expected throughput, right? So if you're expecting to get, roughly like 120 million reads, see, it's much more than that now. For a flow cell, you can sort of calculate it out based on, am I doing paired in 100s or single in 50s, whatever. You basically get a sense of this is how much bulk sequence we're going to get. Let's make an estimation based on. Usually people assume it's going to be some sort of even distribution, though often it's not. And then you sort of make a guess. Sometimes sometimes when people ask me this question, it's kind of strange when they come to me before they go to the actual high throughput sequencing facility people. I often say, depending on what sort of like monetary resources you have, sometimes you might just want to do a test run. That's actually one of the things where also the multiplexing can come in useful. Like you might want to say, like you may not actually care to get a scientifically, statistically relevant number of reads, but you may want to see sort of what your distribution is or how good a particular prep will go. So you might do sort of like an early just sort of test sequencing run, and it will give you some sense of how much you're going to get back, right? And if if you multiplex 10 samples, and you figure you get a certain distribution, and you say, well, if I devote an entire lane to one sample, I should get about 10 times that. We can also tell you, if it's important to people, we can also sort of tell them like what percentage of the reads that, like I said, doesn't always come out to be, equal. It wouldn't always be an equal 1/10. But a lot of these things you kind of have to experiment a bit and then figure out what would be good for the study that you're trying to do. Yes, I mean, yes, I mean that would be kind of the sledgehammer approach. You're just going to say we're going to sequence a lot, and we're going to get a lot of the dominant organism, and we're just going to. But I mean, if there was some way to reduce the amount of representation of the dominant organism, that would probably be the cheapest way of doing it. But if you want to just throw money at the problem, you could just keep sequencing more and more until you get a sufficient number of the less represented species.